Welcome to the Atlantico podcast, where we talk about the science behind the Atlantico project, the Atlantic Ocean, and the human adventures experienced along the way. Here, we have conversations with guests from around the world who work together so that we can better understand, manage, and protect the ocean. So let's embark on the journey of Atlantico and discover the world that lies above and beneath the surface of the beautiful Atlantic Ocean. Welcome back to the podcast. Today we continue our journey into our Atlantic Ocean series, where we discuss one of the scientific topics of Mission Microbiomes of the Tara Ocean Foundation and one of Atlantico's flagship expeditions. I'm happy to welcome Alessandro Tagliabue, who was chief scientist on board Tara as it went to Antarctica. Alessandro is going to tell us about his experience and explain what scientific questions the team has been tackling while at sea. Alessandro, hi and welcome. I cannot wait to hear all about your adventure.
Hi, Eloise. It's nice to be here with you to talk about this exciting expedition.
Great. So before we dive into today's topic, we do like to start the podcast by learning about our guests. So maybe you can tell us about yourself, where your connection with the ocean comes from, and what your journey has been to get where you are today and to what you do.
Okay, sure. So I've always been interested in the sea since I was a child, from my holidays and things like that. So I did an undergraduate degree in marine biology at the University of Newcastle in the United Kingdom. And when I did that, I got very interested towards the end of my degree about the role of iron as this kind of mineral supporting kind of life in the sea, and in particular its role in the Southern Ocean, which is a very, very, very iron poor region, and also a really exciting region of the ocean with lots of charismatic animals living there. So this led me to do a PhD focusing on the Ross Sea, which is part of Antarctica, at Stanford University in the US, where I looked into the controls on the kind of microbiome in the Ross Sea, the influence on the carbon cycle, and things like that. After that, I went to do postdoc positions in Paris for around 4 years and in Cape Town in South Africa for around 2 years. And now I'm a professor at the University of Liverpool in the sunny United Kingdom. And in my work at the moment, I'm really still interested actually in that key question that I was interested in as an undergraduate: what regulates the productivity of the seas? And that's ultimately underpinned by the health and activity of the phytoplankton, these plants that form part of the microbiome. And I try to ask questions in my research about how past or future changes in climate and the ocean environment affects productivity and what the implications of that are for the structure of the marine ecosystem, the ability of the ocean to regulate our climate, and, you know, things that humans maybe care more about, like the marine food supply and the ability of fish to land on your plate. And the main focus of my work is still on these micronutrients, these scarce things that are essential to plants of the sea, and just like they are to us. And yeah, as I said before, this is like really important in the Southern Ocean around Antarctica. So that's what I spend my time worrying about today.
Nice. So you've been going around the Atlantic, you know, from the UK to the US and down to South Africa. You've seen a lot of the Atlantic, I guess.
That's right.
Yeah. And just another quick question. How did you get involved in Atlantico?
I got involved in Atlantico because I worked with the coordinator, our friend Daniele Judicon, and Chris Bowler on a paper that came out of the Tara Oceans expedition about the role of iron in structure in marine ecosystems. And I think that's where our kind of interaction came. And then from that, I think we got involved and started to develop the part of the Atlantico project.
So in today's episode, we want to know more about your experience during one of the topic studies of Mission Microbiomes. You boarded Tara in King George Island in Antarctica, and after days at sea, went back to Punta Arenas in Chile. So to start with, Some big questions. Can you tell us about the region that was studied? What scientific questions were addressed during that leg? Why they are important to address? And then how it will contribute to increasing our knowledge of the Atlantic Ocean and its evolution over time.
Okay, I'll try. So we were interested in the Weddell Sea, which for us, that's one of the two major sort of seas around Antarctica, the other one being the Ross Sea that I mentioned earlier. So the Weddell Sea sits kind of right at the bottom of the Atlantic, southern part of the Atlantic. And for us, we were really interested in, I guess, two parts. The sort of part that is linked to the Antarctic Peninsula. So that's that little sort of finger of Antarctica that points up towards southern Chile and Argentina. And also the offshore regions, so what you might call the Weddell Sea proper, where the water depths are exceeding a few thousands of meters. So the Weddell Sea is really important. It's important for a number of different reasons. It's important for the ecosystems that it hosts, all the penguins and seals and whales and dolphin, all these kinds of things that you might be interested in. It's also a really important hotspot for biodiversity. The marine environment around the Weddell Sea is very, very unique and very atypical. So it hosts rich biodiversity. And it's also a really important region for the carbon cycle, the global carbon cycle. It's one of the few areas in the world where this process of deep water formation occurs. This is where waters get kind of super cooled and super salty due to the formation of sea ice in the winter. So the sea ice, when it forms, rejects the salt. Sea ice is fresh, so it leaves behind cold, salty water, which is very dense, very heavy, and so it sinks into the ocean interior. That means any carbon it's absorbed from the environment gets very, very efficiently transported down into the deep ocean, where it remains locked away from the atmosphere for decades to centuries, even millennia. So it's a really important region for our climate regulation, for our ocean planet biodiversity. And I guess an unusual fact of the Weddell Sea is just as a peculiarity of the sort of currents that go around Antarctica. The part just next to the eastern side of the Antarctic Peninsula is known as Iceberg Alley, because all of the icebergs that carve from the Antarctic ice sheet on the continent get swept around in this clockwise circulation scheme. And so all these icebergs get brought up through the Weddell Sea. So it's a really interesting area to study, but it poses a lot of challenges because we have enormous knowledge gaps in this region about how this environment that I've just explained to you affects the microbiome. And because of these knowledge gaps, we're not really able to monitor, manage, mitigate the impacts of climate change because we're not really confident in the projections and predictions that are made by these Earth system models and assessed by efforts like the IPCC. So to kind of contribute to closing these knowledge gaps, we designed the leg in the Weddell Sea with two main scientific goals, which were all linked to this main underlying question about how gradients in the environment in the Weddell Sea affect the microbiome.
Before you continue, can you maybe explain to us what gradients are?
Oh, changes. So changes in the properties. So Antarctica, the Antarctic seas are not a uniform condition, right? They vary. They vary in terms of the amount of sea ice and icebergs they may have. They vary in terms of the water temperature. They vary in terms of the degree to which that surface layer is mixed. And so the light environment that the microbiome is exposed to, they might occur in shallow water or deep water. That strongly affects the amount of iron that is present and exposed to the microbiome. So there's a lot of variability, but because we haven't really documented impact on the microbiome at the sort of level of detail that is done as part of the Atlantico project, we don't really know, so it's a kind of a known unknown. We know that we don't know this, but until we know it, we can't really quantify its importance in terms of the response of these really important services in the region to climate change.
Good. So back to those two specific scientific questions you were asking in that region.
Yes. So the first one was to try and collect data along a transect. So that just means along a kind of line, effectively, between the shallow waters of the Antarctic Peninsula. So the tip of the Antarctic Peninsula is an island called Joinville Island. That's where we started our our section. And we followed this line effectively offshore. So with the seafloor depth getting progressively deeper, we would hypothesize then the influence of the iron being supplied by the seafloor becoming less and less important until we get out right offshore into the deep waters of the Weddell Sea, where we reached, I think, about 3,000 or 4,000 meters water depth. And the second was so that was almost like a kind of static gradient. You know, we know that gradient exists between the shallow water and the deep water. The second kind of gradient we wanted to sample was a more, let's say, an extreme or stochastic gradient. And that was that we wanted to spend a dedicated period of time following and tracking an iceberg. So an iceberg, as it floats and through the water, obviously it's melting, it's releasing fresh water, it's releasing nutrients, micronutrients present in the ice, because all of these icebergs started on land. And so underneath all that white stuff, there's soil underneath Antarctica. And so as they scour on the soil, they pick up a lot of iron. And then as that iceberg eventually gets, sliced off into the seawater, we think it will be releasing that iron. There could also be microorganisms associated with that sea ice. We really, really don't know. And so we wanted to understand how, especially in this region, as I said before, which is so influenced by icebergs. And also we would expect if as Antarctica warms, the production of icebergs should increase. And we don't really know the influence they're having on the microbiome. So by getting that kind of insight, we hope that we can better arm ourselves with an ability to understand what's going to happen in the future. We don't really know who lives there, what they're doing, how is the diversity and function of the microbial ecosystem adapting and acclimating to these changes in, as I said, iron, light, temperature, sea ice. And, we have a real data desert, which is a bit of a cliche maybe, and a bit of a funny word to use in one of the coldest areas of the planet. But there's a lot of challenges that I'm sure you're aware. I'm sure it's not difficult to illustrate the fact of working in winter and even in summer. It's a very challenging environment. This lack of data means that I guess we don't have the sufficient knowledge, and the knowledge that we gain is really priceless, I think, in terms of our ability to manage the environment, understand how this, pristine, we think, environment is going to adapt to climate change. And it feeds in to lots of international efforts that are going on to try and manage and protect Antarctic environments, like marine protected areas, for example.
Yeah, so it's like trying to find the missing pieces of the jigsaw puzzle. So we have a good view of the Atlantic, especially in the north, and we're trying to find some missing pieces of Antarctica and the southern parts of the Atlantic, so that we can have better models, understand how it works now and how it will evolve over time.
Yes, and I think our understanding sort of decreases as we move away from the north.
Yeah.
And largely that's driven by the difficulties of undertaking sampling. And I guess it's the fact that we can't translate information that we gain from temperate regions to the polar regions. That's a particularly unique problem of Antarctica. Previously Antarctica was attached to South America. And as soon as it detached, this passage opened called Drake Passage. The wind blows from the west there. It spins up an enormous current called Antarctic Circumpolar Current. And this acts as a kind of a geographic barrier. So for millions of years, the organisms that live south of the Antarctic Circumpolar Current have been exchanging very little genetic information with communities from the north. What we know about how phytoplankton, for instance, adapts to changes in iron or light are fundamentally different if you conduct these experiments on temperate plankton that might live in the South Atlantic or the North Atlantic, and Antarctic plankton. So it's the kind of thing where we need to go there to conduct the experiments to collect the knowledge. We can't rely on translating information and transferring information from other latitudes or regions that are maybe slightly easier to get to.
Yeah, so bouncing on that, once you got there in that very difficult region to access, how was that scientific question answered? How did we study the influence of iron and other nutrients and other elements in the water? What did you have to do? And then what will happen next now with those samples and all the data collected?
Yeah, good question. So, of course, you have to get the boat there into the position. Well, first, you have to have an idea of where is the best place to take your samples from to get the best value in terms of answering the questions. And that was a a long effort before we went to sea, actually, to identify places where there was other data sets available, where people had gone to measure similar kinds of things, but not quite everything that we would measure on Tara, where we could ensure that there was the maximum sort of added value for our data. In some places, sometimes it's very good to go somewhere no one's ever been before. But sometimes that actually means that your ability to expand beyond the data that you've collected yourself can be very, very limited. So here we chose kind of not to do that, in the sense that section I spoke about earlier reoccupies a line that the Polarstern, the German vessel, visits every year. Now, they don't do the same depth of sampling that we do on Tara, but this allows us already to have this, to exploit this fantastic wealth of additional information. So once we got to the location, basically we took a range of physical, chemical, and biological measurements. Some of the data is measured using sensors, so sensors that might measure the temperature of the water, or imaging, where you might image the plankton. So that data we get immediately on board, so we can look at it on the boat and make decisions, or just discuss it over a cup of tea. But most of the data that we collect on the boat, we store on the boat and is shipped back to laboratories, and it will be analyzed over the coming months. So it's a long road ahead in terms of, the end of the expedition is really only the end of the kind of first phase of the mission. We've planned a mission, we've executed it. Now we want to obtain the data and put the data together. But maybe to give you an example, some of the data we measure, we measure the temperature, the saltiness of the water. That tells us about how the ocean is sort of layered and how different layers might mix with one another. We can also measure the kind of turbulence of the water, how mixed it is, or how much it is mixing. And that's really important because it allows us to understand how the physical movement of water is connecting different areas that we sample, or sometimes not connecting them, creating barriers between areas that might be actually geographically very, very close together. And we also take water samples. That's a big part of what we do. We, bring liters and liters of water onto the deck of Tara. And then we take samples for chemical analysis. So those include things like measuring the levels of nutrients in the water, the level of iron in the water. We also measure how much carbon is dissolved in the water. And then we can also measure these kind of more esoteric and exotic things like, the isotopes of oxygen. Oxygen exists in different isotopes in seawater. By measuring the isotopes, you can tell whether the fresh water to the less salty water has come from melting of sea ice, melting of an iceberg, or even just snow, so precipitation from the atmosphere. And then obviously we take lots of biological samples, and that, sounds very grand, but in practice, it just involves an enormous amount of filtering of water to try and capture this biological material on filters of different type and different size. All of those filters, they get stored on Tara in liquid nitrogen, generally in different little vials with labels on them. And they will then get they're getting sent back to laboratories and they'll get measured, generally mostly using genomic techniques, as I'm sure other people have spoken about. And, these allow us to get this really interesting and, still for Antarctica, very rare information on kind of who's living there, what genes they have. And importantly, we can measure how the different organisms within the microbiome are trying to transcribe those genes and produce proteins to kind of react to the changes they're perceiving in their environment, which is a really important part of what I'm interested in, is to understand how that environment is linking into the activity of the plankton. We also do some old-fashioned stuff. It's sort of tried and tested, I guess. We use lots of nets, plankton nets, which are probably one of the oldest biological sampling method that you can use on the ocean to collect organisms and to image those organisms, look at them. And so we use those kind of techniques as well.
Yeah, so at one point, all the data, the ones available directly on board and all the ones coming back from the labs on land will come together, and we'll get a better idea of the entire environment and the ecosystems from Antarctica.
Yeah, and I think it's important that that's documents, I guess, what is there. We've also gone there with some quite precise questions, that's how sort of science works. We have hypotheses about how we're expecting certain components of the system to be related to others. And so it'll be really exciting to be able to put those things together and see how well our preconceived ideas match up, or as science usually does, there'll be some surprises in terms of what we find and how different components of the community are reacting, for instance, to the changes we see in the environment. Do you see any unique organisms around the iceberg compared to those elsewhere? Or if you don't, are the ones that are there doing anything differently? How are they reacting to this environment? So I think it's going to be very exciting to start to gain those bits of information.
So hopefully we'll get that information before the end of the project so we can do another podcast, and ask you exactly, so what did happen there?
Let's hope so.
Now to move on to another subject. So each of the scientific studies of the expedition is dedicated to a woman that is involved in marine sciences or was involved in marine sciences. And in this case, it was dedicated to Maria Klinova. So can you tell us a little bit about her and her contribution to the field of marine sciences?
Yes, I can, with pleasure. So Maria Klinova was a Russian scientist. She was a professor at the Shirshov Institute of Oceanology. She was born in the late 18th century and died in 1976, and she was 77. So she was, I think, the first woman to do scientific research around Antarctica. It was quite a difficult thing to ascertain. Lots of women had been in and around Antarctica, but as far as we could gather, she was the first to actually be there for scientific research. So she was there as part of the International Geophysical Year in the late 50s. And interestingly, the Soviet Union was the only nation that participated in the Geophysical Year that included a woman in their expedition. So she was a renowned marine geologist. That was her area of science. Now, I'd say her major scientific contribution was to produce the first atlas of Antarctica. There's a mountain top called the Klinova Peak in the Ellsworth Mountains of Antarctica. There's a crater on Venus, as well as a seamount off Salvador de Bahia that Tara may have gone very close to, actually, that are named in her honor. And the last little anecdote fact I can throw out about Maria Klinova is that her PhD advisor was the Ukrainian scientist Vladimir Vernadsky, who is very famous for developing this concept of the biosphere and this idea that the living organisms can reshape the planets that they live in. And the Ukrainian base on Antarctica is named after him.
Nice.
So she seemed like a good choice.
Yeah, definitely. Very fitting. Very fitting indeed. And, well, I guess to finish on, let's talk about your personal experience on board Tara. Can you maybe tell us what it was like for you to be there and about your life on board and the memories you've brought back from that region of the Atlantic and, of course, of the people that you shared the journey with?
Yes, I'll try to, because obviously there's lots of them. I guess the first thing is that Tara is designed to work at the poles. It used to be called Antarctica. It has a fantastic aluminium double hull. It's designed to be nice and warm inside, and it was. It's nice and toasty inside. I think those working on the boat in the tropics get the counter problem of that. But we benefited from a very nice, comfortable environment inside the boat. Of course, it was a bit different outside. And that was a challenge because much of the scientific work on Tara is undertaken on the deck. There's not really any laboratories inside. So we're outside all the time. And so we have to wear many layers outside, and then you're constantly coming back inside and getting very hot and going back outside. And, working on the deck was hard. It was often windy, snowy, cold. Sometimes it was nice and sunny. And when we got to the farthest south part of the voyage, it was hardly dark at all. So you get very limited, like almost like a twilight for an hour in the middle of the night. But it was tough. The environment was difficult to work on. I mean, Tara performed very well in this environment. But we had some there was a long time where it looked like we wouldn't make it to be able to do this section that I spoke about earlier because the swell and the wind was just too much out there. And that's where Tara is just a 30-meter long schooner. It's not a 100-meter research vessel. And so there are some points where the ocean kind of asserts its superiority over you, and you have to adapt to that. And we did. And eventually we got the weather window and we managed to get out there and do the work we wanted to do. I think the highlight for me, it's the excitement of getting up every day and going out on deck and seeing what the conditions are like. Personally, I spent over a year planning this expedition. So thinking very carefully about where we'd collect the data from these particular locations. So, it's a kind of point of principle and excitement to be the first out on deck in the morning to look and see, where are we? Where is this place that existed up until then on a map or a set of coordinates? So that was great. And as an oceanographer, that's what we get really excited about, getting out on the ocean. And you want to make the most of everything. I think a real highlight was, and something very unusual about Tara, was the ability to, when we had some downtime, to go ashore and to take the Zodiac, go on to the Antarctic continent, wander around, have a look at some of the flora and fauna that are existing there. And that was very interesting. You've got a lot of human influence, despite the fact that I said it was a pristine environment earlier. This part of Antarctica, the Antarctic Peninsula, it's been used by humans for a long, long time, from the days of the whaling expeditions and things like that. So there's actually a lot of traces of human activity on Antarctica, which is quite interesting, I think, to experience, alongside the kind of solitude. And experiencing both on land was actually really, really cool. I think a real highlight was when we were waiting to cross back to Chile, to cross Drake Passage. And that's a very, anyone who knows the sort of ocean will know that Drake Passage is a very windy place. That's where that wind that drives the current that I spoke about earlier, that's where it passes. And so it's very, very windy. And if that wind is not coming from the southwest to west, it was not going to be possible for us to cross. And so for the last part of the expedition, actually kind of trying to guess and estimate when that would be and making sure we weren't a long way away from where we needed to be at that time was a bit of a challenge. So we were anchored in Deception Island, which is an old volcanic caldera that has collapsed. So it looks a bit like a horseshoe. So you can sail inside the caldera. And so we anchored ourselves up there, waiting, and tied everything down for the Drake Passage run. And then managed to go ashore and spent a fantastic afternoon hiking on the volcanic kind of peaks there, which was really unique. I think you're not gonna get to do that in any other way, really. So that was really fantastic. And I think even since I was a child, I've always wondered about this strange named island, Deception Island.
Deception, but you were not. There was no deception there.
Not to my knowledge. Yeah. So, but it was really great, and I think. I mentioned about the tough conditions that we had, and it was difficult. There were times when you finish your day's work and the boat is still bouncing around everywhere, and you need to try and sleep, and you can't sleep very well. You're putting clothes underneath your mattress to pin yourself against the side of the wall so that you don't roll around too much, especially for those of us in the top bunk. But it also generates, this spirit of collaboration on board the boat amongst everybody sharing in this sort of shared endeavor, this shared adventure to try and achieve a set of goals, to experience this environment together. And that fits into a long spirit of Antarctic collaboration, I would say, that really is quite common to the international scientific community who work down there. So we had in this voyage, lots of collaborations with scientists from the British Antarctic Survey who helped us with the design of the iceberg experiment. We had developed this very nice collaboration with German colleagues on Polarstern, and indeed Polarstern occupied this section that we did again around a month later, and took the same type of genomic analyses on there because we developed this coordination with them. We also had these links with Spanish scientists on the Hesperides. And, it's that spirit of collaboration, both within the team on Tara and amongst the wider scientific community, which I'd really like to kind of emphasize as being maybe the best part of the science that we do.
That sounds amazing. And we'll put in the show notes, we'll put the link to the videos that were made during that leg, because they are amazing. And the pictures that were taken are just beautiful pictures, and penguins on that slope on an iceberg, which is,
that was crazy.
It was amazing.
Because when we sailed towards that, Eloise, we could see this iceberg, and we thought these two icebergs that were next to each other, and they looked interesting. So we were going that way anyway. So we sort of just detoured a little bit along the same trajectory, and we came from the back of that iceberg actually, and so you could see at first a couple of things on the top of the iceberg, and then it was only when we kind of rounded the corner that we saw that there was the whole slope of the iceberg was just full of this group of gentoo and Adelie penguins kind of sliding down, jumping in the water, trying to get back on. And that's, we're all scientists, we're all serious about what we do, and we are serious about what we do when we get out on the ocean. We're there to do a job, but at the same time, everyone will stop what they're doing and enjoy this natural spectacle.
For sure,
which we're very privileged to be able to do.
For sure, you have to enjoy and that experience that is so unique. I live it through your words, but I imagine it was a wonderful experience.
It was great.
Excellent. So I have to thank you for your time today, for sharing your insight and that exciting adventure in that cold environment and difficult environment. And as I mentioned earlier, hopefully we'll get all the analysis done soon enough so that we can have a discussion again and actually talk about that microbiome and the ecosystem in that region and see what we discovered.
Brilliant. That'll be a pleasure. Thank you, Eloise, for having me.
Thank you again. And for those listening, just keep coming back to learn more about other regions of the Atlantic. We'll be back soon. We hope that you've enjoyed today's episode and look forward to seeing you next time. You can follow the Atlantico project on our website on www.atlantico.eu, and you can also find us on Twitter, Instagram and Facebook. All the links and information on the project and on today's episode is in the show notes. Atlantico is a project funded under Horizon 2020, a European Union research and innovation programme.